I will go ahead and talk about what we've been doing using UVC to suppress fire blight on apples. A little bit of background about the process. Come on, mouse. There it is. Yep, I'm going to minimize that a little bit. And yeah, germicidal light's been used for a variety of different things. It's very effective against transparent organisms or organisms that love being photosynthetic. Transparent bacteria, algae, it's used in medicine, aquaculture, agriculture. I use it in my reef tank at home. And that's what made me start to think it could actually work. Once I've got rid of my coral bleaching diseases, I was thinking, maybe this could actually work on something like fire blight. It is allowed in organic agriculture. It leaves no residue. What does it simply do? It causes these thymine dimers in the DNA helix. Here's an incoming UV photon. Can you hit share? Oh, it's not sharing?
It's looking good, Kurt. We can't see you, but your slides look great.
Oh, okay. Gotcha. All right, so you can still see the slides. Good? All right, good. I cut that. Excellent. Yeah, it causes these thymine to thymine dimers. I'll turn my head on during the questions. How about that? I think I've got too many video things going. Little photon comes in, pops it, the two TT base pairs bind, and then that makes a mess of the organism's ability to divide and do all the things that it needs to do in terms of making proteins and stuff like that. Sadly enough, many organisms have a photolyase-mediated repair, so you can shine blue light back in on these. The enzymes are activated by blue-violet light, and they can just fix these base pair damages, which is wonderful for us if you like to wear those blue masks that you see people using in alternative healthcare things. So that blue light actually can fix UV damage, particularly in organisms. Unfortunately, Erwinia has this as well, and I'll talk about that. Early uses really focused on CEA and indoor production, like in Norway, greenhouse strawberries, greenhouse other plants, because in the greenhouse, powdery mildew is sort of the king of the diseases, if you will. And we've just now started beginning to use this in apple as sort of a proof of concept. We've been 2 generations in on equipment. This is the newest one. The 3rd generation is the one where I actually talked to the engineer myself because David Gaduri spoke on my behalf. And then we got 2, one grape size model and one larger grape size model. Consequently, David used to do a lot of work on grape, used to manage powdery mildew on grape and strawberry. Here's some beautiful pictures of grape powdery mildew from David's work, a little bit of powdery mildew on the leaf. And as you can see, powdery mildew is transparent and would easily be susceptible to these types of diseases. This types of technology. The UVC light will go right in and get it, if you will. And then there's always a little bit of trying to balance the host tolerance for UV and the dose needed to kill the pest or pathogen. Oh no, I hate that. I just heard something disconnect. Let's hope you're still there. The most intolerant to UVC is probably something like hemp, and then least is grape, and then strawberry is very not really affected by UV radiation whatsoever. You can just sort of zap these things. This is a picture from Florida where they're doing this types of stuff in Florida as well. And this is where we first started. We grabbed David Gaduri's grape model. I reduced the trellis on one of my Evercrisp plantings, and then we started going. We spent some time in the lab, and what's shown here are the mean EA, log base 10, CFUs per flower on and by dose of UVC radiation. And you can see if we incubate it in the dark, the Erwinia amylavera has the photolyase-mediated repair enzymes. You can find the genes of these reported in paper. So anyway, we first attempted it with a grape writing unit. Is it back or is it gone?
It looks good, sorry, you're fine.
Okay, good, just double checking. Yep, so we used a grape unit made for grapes. We dropped a trellis, did this on Evercrisp apples, and then we measured our colony CFUs after zapping flowers inoculated with 10 to the 6 Erwinia amylavera. And you can see pretty quickly that you can wipe these things out at night with about 200 joules. But that's because the photolyase-mediated repair doesn't occur at night. It needs that blue-violet light. Turn this off, and then you can kill the Erwinia much more quickly at night. And then, of course, I've learned after spraying all my own orchards in the day, going back at night is terribly unfun. So we like, what dose do we need to make this thing get cooked during the day? Looks like about 400 is enough to kind of like burn these things off the flowers, if you will. And so what we were thinking is we will start to move this to the field. Infections happen at bloom that start the fire blight epidemics, lead to the awful shoot blight that ends up devastating your small plantings, particularly of high density apples and all the ooze and whatnot. And then, you know, in general, it's challenging to get those antibiotics on at just the right time. We have antibiotic resistance, they lack consumer appeal. And then I thought, if these things are multiplying on the stigmatic surface of the flower, I'm going to try to use the laser pointer, then perhaps this has an opportunity to get it. It could work well on a natural inoculum, transparent organisms, on organisms like Erwinia that's right on the surface of the flower. This is the newer unit. And what these do with the little reflectors is it kind of bounces the UV light around to create a cloud of UV, if you will, because these flowers are not always oriented face out towards the unit, depending on the types of planting. So we grabbed this site. It was a high-density super spindle planting. Now it's a mess. It's Evercrisp. It's an apple out of the, I guess, Ohio breeding program. Maya G41 2019 dropped the trellis down to 96 inches to accommodate the silly grape unit, and then we did replicate plot panels, and these are the types of treatments we put in. We did streptomycin at 80% bloom, put that down, put down Blossom Protect with Buffer Protect because we know it's really good. Then I dropped the hammer of 10 to the 6 CFUs per mil, EA273 that evening. Checking, I heard the watch vibrate. That evening I went back, zapped it with 200 joules, and then I went back again with strep and Aurea basidium and the UV, unfortunately, the next night. And those got right on in the evening, that got on at night. And then I measured blossom blight, which is what we were targeting, and the ventral shoot blight incidence. We also grabbed EA populations, leaf shape, shoot growth, because that's where we would see potential UV injury. At this point in time, we didn't know how apples were going to respond. 2021 was an exceptionally cold bloom. Low levels of infections on Evercrest. This is what the graph looks like. It's kind of complicated. This is the mean incidence of blighted blossoms, and this is the mean EA log 10 CFUs per flowers. That's going to be in purple. Blossom and shoot blight are going to be in blue and green. We did get a mild reduction in the Erwinia populations, similar to firewall. And as Quan has published and shown many times, the Blossom Protect doesn't often reduce populations in the flowers. So nice that it supports his research just serendipitously, and someone can repeat it for him. Great. Excellent control, pretty decent of blossom blight, shoot blight. It was a cold year. It's not going to be a challenge. This is blossom blight, shoot blight, firewall, blossom protect. UV is not as good, not statistically different. Now, 2022 was an awful year. Nearly the entire Cornell Agritech experiment station lost all its apple plantings. So this was a real beast, warm, wet, high levels of infection on Evercrisp. That crispy picture I showed of the block is what happened at the end of the summer. The whole thing died because it's a small high density planting and everything died. But during the time when we used the unit and the strep and the blossom protect, really pushing about 80% blossom blight this time, 40% last time, 40% shoot blight. This was a really devastating year. Still get a slight drop in the populations on the flowers with firewall and UVC, but things are going to bounce back, and those populations are much higher than they were in 2021. No real benefit to using Blossom Protect on populations, but the shoot blight and the blossom blight being in blue, shoot blight being in green, look pretty good, all things considered, with all of them. Not as strong with the UVC, but then again, we're really shining a light on flowers twice, so it did show a little bit of promise. Still high on, but a little better in a really nasty year. So the other thing that we needed to do in order to make sure this works is check and see if there's any differences in the leaf shape and internode length. The leaf shape and internode length are where UVC will first start to show its damage to the crop. And what we have here is leaf length, leaf width, internode length on first year shoots. If it gets damaged, it'll have really shortened internodes. I did 2 inoculum doses in the early on, but really only showing 1 since there was really no difference. I did a 10 to the 2 and a 10 to the 6, but what we showed today was 10 to the 6. Untreated, streptomycin, aureus basidium, and 200 joules per meter squared. Didn't really matter, didn't seem to affect it. 2022, no significant differences in shape. And then growers are still paranoid and worried, so we continued our studies. And I had extra trees left over from our scree defense inducer trial. So these were out there. Yeah, they got wrecked pretty hard in 2022, but they're still trees. And we continue to do different things to them. Did I inoculate them? Not in the way except for what was required by that grant. Trellis was already pretty small in that particular planting. It never got very big. And then we did them in sort of randomized complete blocks with 5 trees per plot. And then I did the same kind of thing in these extra bonus trees. We had 80%, 100% bloom, streptomycin, aureus basidium. I didn't do any inoculation with EA273, and that trial was inoculated further down the road, so there's a little bit there. And then what I wanted to do was weekly and bi-weekly applications of UVC. The growers were worried that these could impact the horticulture of the crop. I mean, this is already a devastated planting, so it's kind of weak sauce. But we wanted to at least see if the leaf shape in the internodes would be changed. We didn't really get a lot of blossom and shoot blight incidents in this particular section of the planting, and we looked at the populations, leaf shape, and tree growth. Really didn't see anything overall, depending on if you look at Gala or some of the others I'll show. You can pick up low levels of EA in the untreateds, but the other things really didn't have much. Whether it was UVC once a week or twice a week. Canopy height is pretty terrible for these crops because they're devastated in general from the 2022 massacre. But for the most part, they're just small, runty little trees at this point, and no real difference. The UV didn't seem to be impacting them. We got some on Lady in Red as well. Same trends, no real differences in length, width, internode length, or first year shoot, or canopy height. Small trees, but no big deal. Same with Royal Red Honeycrisp on the few trees we had of that as well. So we felt that in the end you could get some control, about 200 joules per meter squared against high inoculum. What level of dose is this? This is the same dose that it would take to kill powdery mildew in strawberry and grape. Sometimes people might go as high as 400 in a field, but we've had cold and warm bloom seasons. We've had fairly comparable results compared to strep and organic, and reduced the populations on the surface pretty okay. No apparent damage or no impacts on the development of young trees. The nice thing about it, no residue, no potential for, and there is potential for frequent use in robotic automation. If you're not a poor Cornell scientist and didn't have to do everything yourself, you could have robots and robotic tractors pull this thing around, and you could even clean the flowers off and then apply Blossom Protect on after it, or something of that, if you really wanted to go whole ham on organic fire blight. Georgia's done some studies with cleaning the niche, and maybe they don't always work out, but if it's just using lights that you already have on your tractor, why not? And then we did a little bit, we shifted gears from fire blight briefly, but this is going to come back to fire blight pretty quickly. Like, what? Fungal diseases on apple? Who cares about that? This is a fire blight meeting. Well, we often wondered what would happen to apple scab if I ran it all summer long, and we're not going to spend a lot on this. We noticed they can tolerate applications, and is there impact on other diseases? Well, Venturi is so darkly pigmented, it can't do anything. It also has photolyase. It's going to need big doses. Well, that's not the subject of this talk. We did look at big doses. We started doing these trials on a different planting of Evercrest that I planted later at the end of that 2022 massacre because everything died. Dropped the trellis back down for our silly unit, did the plots, and I did, untreated. I did a full Captan Mankozev, rotated with fancy single sites and then double nickel, and rotated weekly with other single sites. Did UVC 200 at night, 400 during the day, and I timed everything using a combination of RimPro, but that's not why we're here. What I wanted to see is what happened to fire blight in this block when I used a season-long UVC application program. If apples end up getting repeat applications for fungal disease, is there any impact on seasonal fire blight development? If we go back, you'll see that there's no fire blight treatment in here. Well, double nickel on treatment 2 has a potential for fire blight, but nothing else was getting any fire blight except the UV programs. And so I wanted to kind of look and see what was happening. And then I've often wondered for the longest periods of time, did the UVC, can it do anything to an ooze droplet at all? You never know. What about if I put down kasugamycin and I zap my trees with UVC? Am I going to break down the kasugamycin? These are the questions that I wanted to start picking at and answering. So we've been doing this trial for scab weekly all the way to harvest in the Evercrisp, and I wanted to look at what did the tree mortality look like after 3 years of the study. And so this is what we have. We just picked up the data. We don't have the apple scab data for 2025, but you know, this is a fire blight meeting. No one cares about apple scab. So the mortality is pretty high in the untreated blocks. They pretty much got wiped out. The biological ones really didn't do much. And you're like, wow. Even some of these fungicides at some level must have had some impact on the fire blight bacteria, but over time, the ones where we zapped 200 during the night, 400 during the day, it must have been doing just enough on the natural levels of fire blight that showed up to kind of save the day. Now, we're just now starting to see shoot blight develop in this block because it is 96 degrees and 100% relative humidity, and the shoots are still growing in Agritech. And so I took a little bit of data on this. Nothing is happening yet, just little snippets here and there, and doesn't seem to be doing too much on natural shoot blight development in this particular year. But the other thing that I really got interested about was trying to see if this could do anything to an ooze droplet. So we collected newly formed ooze droplets and blocked the ooze droplet by size and color. Ooze droplets can be a range of different colors from amber to white. You can see an orange one right there. Some of them are semi-transparent. And so we put them, made sure that those things were sort of put into their blocks so we could use them as kind of random effects in the model, whether it's size and color, or we could correct for size at least. We know colors are a little different, and then I assumed that these things would be very recalcitrant because they're just sugar and compacted bacteria, and so we started zapping them with high levels of dose. Each time you run through the field, the dose is mildly additive, and so over time, you might get some impact on these things, and then we compared it to streptomycin. I didn't expect any real impact to the ooze droplets, but there might be a little. And this is where our experimental setup happens. We ran out to the field, we collected the ooze droplets, we isolated them, hot glued them to plates, quickly measured them, and zapped them, and then processed them for colony counts. We did 2 different ways. They were very recalcitrant. Sometimes they're sticky, but they can take about 30 minutes to dissolve and buffer, or more, with vortexing. And then we thought, we'll dissolve them fully. And then, like, well, what if a scenario like this occurs? If the ooze droplet is sticky and it rains on it, it's not going to sit in a vortex for 30 minutes until it dissolves. It might rain. So then we picked the ooze droplets off, and then we washed them, and then removed the ooze droplet. The idea being, I wonder if, say this was an ooze and not a piece of bubble gum, if the UV hits it, will it bake it like it's burning my skin and making a scab so that the droplet becomes more of a jelly bean instead of a sticky piece of bubble gum? As we started playing with them, they're initially very sticky until you bring them in and freeze them down, dry them out, and then they become more like jelly beans. And so I was like, maybe we'll wash them, see if we can lock away the bacteria in a scab of sugar, sort of hardened by UV, like we're curing candy, and then maybe that could prevent or reduce spread. So we did 6 replicates, and then we repeated the study twice. As we all well know, oozes are very high on bacteria. Mean log CFU divided by the square micrometers of ooze droplet. And for the most part, these things are fairly recalcitrant. Depending on the study, if we fully dissolve an ooze droplet, the candy coating goes away, just loaded with bacteria. No big surprises. Not even strep can really beat these things down. And if it did, it would take a huge dose. If we rinsed them for 30 seconds in a vortex just to see what was still active on the surface, we could start to see some little bits of impact, with the caveat that this stuff is fairly variable. 1200, a slight reduction from, log 10.6 versus log 10 4.5. And then strep actually does a good bit of killing some of the residual bacteria that's more surface active on the rinsed ooze. So we're sort of just repeated that a couple of times. And, I don't expect anyone to intentionally use UV for ooze, but if you did, maybe something good would happen and you could make your ooze droplets jelly beans instead of sticky bubblegum. The other thing we wanted to do was look at what does it do to the antibiotics. This is the final thing we ended up looking at. We took a bunch of flowers that were treated and or plates, CG plus rifampicin, and amended with 100, 200 streptomycin and kasugamycin. We're using CG plus rif because I put on EA110R. We then exposed everything to 1 to 300 joules, 3,200 joules, which is at a ridiculous dose that you would probably never use in the field, even though we did use it on oozes. And then we plated it all. We took EA110, dilution drop plated it, and then checked the colonies. And for the most part, 10 replicates repeated. We didn't really find an impact of UVC. I think the bigger problem with something like kasugamycin is just daily exposure to the sun throughout the day in terms of breaking down. Zapping these things for intense doses didn't really impact the ability of the antibiotic to be present. I mean, this is the plate where there was nothing on it, but the other plates are a bunch of empty CG plates. It's not really that interesting to look at. So, sort of wrapping things up. Hopefully you're still there. Yep, season-long use of UV in 204 had an impact on mortality. For the season-long stuff, orchards receiving only fungicide management, application of ooze droplets could harden the surface a bit, cure that candy coating on the ooze droplet to reduce the potential for rain dispersal. And even at extreme doses, doesn't seem to impact antibiotic residues. I think we have enough to worry about that from normal breakdown. Overall, it seems effective for fungal and bacterial pathogens. You can use it in organic. You will need to apply pesticides for other issues. And what do? Run tractors day and night? I don't know. The other thing we've often worried about a little bit, and everyone brings it up, is are we accelerating antimicrobial resistance in non-target fungi? It's not something we've looked at yet. Stuff like botrytis is known to mutate frequently and have resistance to everything. And this is Candida auris. You don't need to tell you what that is. It's bad news. And then our growers ask, how do we move forward? I think we need a consistent performance from a design that apple growers can use. This is not the most efficient design, even though now that I get to talk to the engineer, I'm going to make one that's 3 to 4 more feet taller and wider. Terrence does have an over-the-road sprayer. I kind of want to model it after that and give that a shot. This is a lot easier for growers to use, but we know that the open design is more impacted by temperature and cooling environments. This is more shielded. You lose a lot of the reflection here, and you really have to get kind of close. You do need a fruiting wall. I think the technique is probably best suited for nursery or fruiting wall. Probably a nursery situation where you're going to baby the crops and spray them a lot anyway. You could drive this around at night. We're getting 3 years of data. I'd still feel better if my colleague Scott Cosboom had gotten some more data out of his area. And I'm going to go ahead and wrap it up. These are my people. Everything in my lab is funded by growers and commodity organizations.